Hi, everyone. This is Neil Littman. I'm here today with Eric Schur, who's the founder and CEO of HepaTx. BioVerge is looking to collaborate with HepaTx. They have some pretty exciting technology that they're developing for a variety of liver diseases, starting with acute liver failure. So I will turn the presentation over to Eric to walk through a short pitch, and then we'll save some time for Q&A at the end. So Eric, without further ado, I will turn the presentation over to you to screen share and go through the deck.
Thank you very much, Neil. Let me share my screen and we can talk a little bit about HepaTx. So this is the team of HepaTx. There's myself. I've got 25 plus years of experience in the biotech industry after I finished my PhD, and my co-founder Mark has got both of his degrees from Stanford, coming out of Irv Weissman's lab. We met at Stanford developing a commercialization plan for the technology that has become HepaTx. Gary Peltz, the inventor of the technology, is a professor of anesthesia at Stanford down in the lower left, along with our other advisors along the bottom and our operating team in the middle. And the problem we're addressing is late stage liver disease. Everybody identifies with the fact that there are not enough organs for transplant for people who can use them. But that problem is particularly large with late stage liver disease. In the US alone, over 200,000 people each year find out that they've got late stage liver disease. We spend $10 billion plus on those transplants and just keeping the remaining people alive. Transplant is the only efficacious therapy for these people. So in order to address that gap, one strategy is to transplant actually liver cells from these patients, kind of analogous to a bone marrow transplant. But the drawback to that approach is that those liver cells are coming from the same livers that are being used for transplant, so they're in short supply as well. Alternatives like differentiating stem cells to hepatocytes have been attempted, and so far those have all failed. But the SF-HEPS technology that we licensed from Stanford and are developing, we think enables that strategy to differentiate stem cells to the hepatocyte for transplantation and enable us to fill that gap. What we're doing is shown on this slide. We take fat tissue from normal donors from liposuction procedures, which is a very rich source of stem cells, and right now is just discarded. We can readily isolate those cells, expand them, and then use the technology we're developing to differentiate those cells into hepatocyte-like cells in culture. Those cells can then be infused into patients' circulation. They'll home to the liver and replace lost liver function. We've done a lot of work developing this process. So in our lab near Stanford, we've dialed in the process for making these cells so that we can repeatedly make these cells, and they come out the same way each time. So the next several slides have some data demonstrating the characteristic of these cells. For example, these data are RNA sequencing data that show that our hepatocyte-like cells, SF-HEPs, come out expressing the right kinds of genes that you'd expect in hepatocytes, all the way from lipid metabolism and urea synthesis down through drug transporters. In addition, these cells synthesize a number of cytokines that they secrete, which would be very useful in liver failure patients who have a rather inflammatory condition. And several of these are anti-inflammatory cytokines, and others are trophic cytokines or growth factors, basically, that can help their liver heal. And then finally, these cells possess the right kind of hepatocyte functional properties. They're able to transport bile acids like normal hepatocytes. They can synthesize and store glycogen, and they can also uptake and process LDL like normal hepatocytes. So we've got a great in vitro property of these cells that shows that we've made them into hepatocytes. We've also put them into mouse models of acute liver failure. The model here is one in which we have cloned a gene into these mice that allows us to give them a drug to basically give them acute liver failure. So when we do that, we're then able to infuse our SF-HEP cells into these mice via their portal circulation, then subsequently test them. In this slide here, what we're seeing in the top row are SF-HEPs that have been transplanted into this mouse. This is sections of the mouse's liver after 2 months after transplant, looking at an immunostain for the human versions of these proteins, albumin, CK818, and ASGR1, demonstrating that these cells are engrafted in this mouse's liver, and so this mouse has a human liver cell graft in its liver. The bottom row is a mouse that was infused with the starting stem cells, and what that demonstrates is those stem cells don't spuriously integrate and differentiate into hepatocyte-like cells in mice. We've also looked at these mice over a longer term. So here in this slide, we're looking at synthesis of albumin from those engrafted cells over 2 months. So looking at for human albumin in the bloodstream of these mice, we can see that over a period of 8 weeks, there's increasing amounts of albumin synthesized and secreted in these mice. And that amount of albumin reaches 0.8 milligrams per mil. So the two things we can take home from this slide are, one, that these cells persist as an engraftment, and second, that 0.8 milligrams per mil is actually a clinically significant amount of albumin being synthesized and corresponds to about a 10 or 20% replacement of the liver. And finally, we've simulated a clinical trial in these mice, the same TK-NOG model. And when we treat them with that drug to induce acute liver failure, as shown in the gray line, if we don't treat them with anything besides that drug, they die in 3 weeks. However, if we give them that lethal dose of drug and then treat them with our SF-HEP cells, as shown in the orange line, we increase survival to 40%. And if we could achieve this result in humans, we'd be very happy about it. Our clinical plan for these cells is shown in this slide here. Our first in human population will be patients with acute liver failure due to alcohol ingestion. And that's for a couple of reasons. First, these patients have a very high unmet medical need. About 30% of these patients die within the first 30 days after they're admitted to the hospital. Second, that these patients are generally not eligible for transplant, so their fate kind of lies with the gods, so to speak. They hopefully will get well, but there's no other treatment to offer them. Since they have acute liver failure and they don't have a long history of drinking, the barrier to engraftment of our cells in these patients is lower, and we're more likely to see a clinical success. So with success in this patient population, we'll be able to then further develop these cells and address the larger population due to chronic liver failure. Our go-to-market strategy is efficient. We spent a lot of time thinking about how to take our product from where we are now to get it to patients as an approved product. Two aspects to that are manufacturing and our clinical and regulatory strategy. So in manufacturing, one of the things that Mark and I really liked about this technology was it's very scalable and differentiated these cells into hepatocytes quickly. So we are able to make literally liters of these cells, and we can do this on a human scale. In addition, the components of the manufacturing process are already available in a GMP type of format. And finally, we don't have to do tissue type matching with these cells just because of the nature of hepatocytes. We're able to simply do blood group matching, which means we can make 3 products and cover nearly the entire population. Together, these items will make manufacturing much more simple and reduce the cost of goods. On the clinical regulatory side, the reason why we chose that acute liver failure indication as a first-in-human indication is because we can get an orphan disease designation with it. What that allows us to do with the high unmet medical need is if we have a successful phase 1 trial, we have the potential to bridge that into a phase 2 trial, which if that trial is successful, could lead to an approval at the FDA. So In short, we could potentially get an approval on as few as 2 clinical trials. The differentiators to this approach are shown on this slide. First, we've broken that supply limitation. We essentially can make an unlimited amount of this material. From a single liter of lipoaspirate, we can make enough of these cells to treat 20 patients at our current scale, and we expect process improvements to increase that number. Second, we can do this as an off-the-shelf therapeutic. So we can manufacture these cells, bank them, and ship them for use on demand. And finally, we look at it as a broad spectrum therapy. This should be applicable to not only acute liver failure, but chronic liver failure from a variety of indications, allowing us to make a single product that can address a very large market. Our timeline is to get to our initial IND filing within 18 to 24 months. The clinical trial that we have designed runs about 1 year, so we aim to have our first clinical result in humans in 3 years. So just to recap, it's a very large market without any compelling solution right now. We're developing a simple cellular infusion to treat many of these patients. And we've developed a very efficient go-to-market strategy. We've got great proof of concept both in vitro and in vivo, and we've got a great team working on it. So we hope you'll join us.
Thanks, Eric. That was great. A couple questions to dig into. One question I always like to start with is, how did you assemble your founding team and your advisors?
So Mark and I had met at a program at Stanford to develop a commercialization strategy for one of the 1,800 technologies that Stanford had available. And so we could take our choice. And Mark and I both have a stem cell background, and we identified this technology as one that we thought was eminently commercializable, had a big need that it could address, and the inventor was somebody who we got along with. So that's how we met and got started with Gary Peltz in founding HepaTx. Once we got the ball rolling with HepaTx, we identified advisors who could really address key needs that we'd have in development. Gary, of course, is a liver biologist and geneticist, and has, of course, extensive work in humanizing livers and doing liver cell transplants. So he was key. Gary, in turn, introduced us to Marie Chetta, who used to be CERM's head scientist and has a long history as both a stem cell biologist and a liver transplant physician. And then finally, Shannon Dahl is a remarkable person who, out of her graduate work, co-founded Humacyte, which is a company developing artificial vascular grafts, which are now in phase 3 clinical trials to help patients with dialysis issues. And so she's got a tremendous amount of experience going all the way from the bench through regulatory, and in fact got the first RMAT designation from the FDA for her company, Humacyte.
And the RMAT designation, actually, maybe we could just spend a minute on that. Is that something that you would anticipate applying for from the FDA at the appropriate time?
Yes, I think that's certainly an option. That and the orphan disease designation are the two that we talk about, but they both offer us an opportunity to accelerate our development and get some preferential treatment at the agency.
Right, and just for the viewers, RMAT is the Regenerative Medicine Advanced Therapy designation. So it's another tool for accelerated development, in this case for specifically regenerative medicine based therapies, which obviously HepaTx would fall into that bucket. Okay, that's great. So maybe we could talk a little bit about your long-term scientific vision. So you've touched upon this throughout the presentation, but fast forward 5 or 10 years into the future, what is your ultimate vision for the technology that you're developing?
So liver disease is a huge market, and it would be a wonderful advance if we can help address some of the problems there. We anticipate this first product to be applicable in acute liver failure, and certainly we'll move it to chronic liver failure as well if testing indicates that it should be. But we, in addition, are developing a platform to develop additional cell therapies, both for liver failure as well as other disease states. So in the long term, we anticipate doing not only a single liver product, but additional liver products and cell therapies for additional conditions.
Great. And then as you're moving forward to execute across that vision, what do you see as your sort of biggest challenges in both the near term and longer term? You know, obviously there have been a lot of attempts to develop hepatocyte-like cells. There's often challenges in how they actually function or in they graft. So just out of curiosity, what do you see as your sort of biggest near-term challenges? And then if you're able to overcome them, what do you see as some of your longer-term challenges that need to be overcome?
Yeah, so really our near-term challenge is in vivo assessment of these products. There are the animal models which are required in order to test these in humans. I think we all want that to happen. No single one really tells you the right answer, and they're time-consuming and expensive. And so a challenge is to really assemble those data in a format that can convince scientists that there is a likelihood to be efficacy in humans as well. And so in the near term, we're really looking at trying to optimize our ability to do in vivo testing. Translating that into humans, of course, is always a challenge with every therapeutic program. So that going on into humans will be the next risky area for us. And we do everything we can to anticipate those risks ahead of time, but that's always a point at which we're, we can't anticipate the outcome completely.
Just a couple questions there. Just in terms of translating this into the clinic, so you touched upon animal models. What is the robustness of the animal models for acute liver failure in particular, since that's your first indication? How well do they recapitulate the human disease? How well do you expect them to translate into clinical results?
We're approaching this with the strategy of using more than one model, because each model kind of tells us a part of the story. And so the model we've been using so far has been one of acute liver failure, which is quite good. It's one of the best out there, but the mouse is also very immunocompromised. And so we're looking to adopt a couple other models, one that recapitulates some of the features of chronic liver disease in a mouse that also has an immune system, so we can factor that into the equation. And then a model of acute alcoholic hepatitis in mice, again with an immune system, so that we can factor in aspects of that liver disease into our analysis as well. And both of those latter models are considered to be a little closer to the human case and be more reflective of what we're likely to see in humans.
Great. And Eric, you touched on an important point there. I just want to dig into a little bit. So you mentioned some of the models you're using are in immunocompromised animals. As you fast forward to the clinic, as you think about your target product profile, could you talk just a little bit about your TPP and then the role that immunosuppressive drugs might play for patients who do receive these cells over sort of, let's say, not just acute, but in the course of chronic treatment?
Yeah, so the way we think that these cells will be applied is via a portal circulation infusion from an interventional radiologist, which has been demonstrated to be reasonably safe in humans for acute liver failure initially, and then we plan to go to more chronic liver failure as we further develop the product. We expect that we will need to use some type of immunosuppression just based on our discussions with transplant surgeons in designing that initial clinical trial. However, one fortunate fact is that the liver and liver cells are not particularly immunogenic, so the level of immunosuppression should be relatively modest. And it's the same strategy that they use for the whole organ transplant is what we're going to follow. So we have no reason to expect that that will create problems over the long term for patients. So, in short, we're looking at an infusion of cells to treat a variety of liver disease, which will require probably some immunosuppression over the longer term for patients.
Great, thank you. And then finally, my last question, it's probably relatively obvious by now, but how do you think this impacts patients with liver failure, whether that's acute or chronic over the long term?
How do I think it
Well,
impacts
well, it
patients?
will impact patient lives essentially.
Yeah, so in the near term, the patients that we're treating, as I mentioned, are one of our hepatologists had an example, and it was a patient who came to him from Arizona, and she was like 23 years old, and she has liver failure, and he has nothing he can do for her. And so it's really up to chance whether she's going to survive or not. And so we think that this therapy will be able to give some of those patients a shot at living longer than 2 weeks. And so certainly, it can rescue people who otherwise would die in the near term. We think in the longer term, with appropriate development, that this may be able to help people with more chronic liver disease who are repeatedly showing up in the ER and doctor's offices with bacterial peritonitis and esophageal varices, to come back to a situation where their liver function is sufficient for their body and to lead a more normal, healthy life.
Great, thank you, Eric. Well, with that, that's all the questions I have. So I think we'll wrap up, and I'd like to thank Eric for providing an overview of HepaTx and the very exciting technology that they're developing. So thank you, Eric.
Yeah, thank you, Neil. I really appreciate it.